So welcome to the 7th Anne Klibanski Visiting Lecture. My name is Miriam Bradella. I'm the director of the Center for Faculty Development, and it's great to welcome our Dutch colleagues from Rotterdam and Erasmus University and our MGH faculty here. So before we start the main lecture, I would like to say a few words about the Anne Klibanski Visiting Scholars Award, which was established to support our women faculty and postdoc during the COVID-19 pandemic. And this award allows them to serve as a virtual visiting professor at a national or international institution, and that's organized by the Center for Faculty Development. We selected 36 amazing women, which you see here. I want to highlight Meenal, who's going to talk today. And they were giving presentations really much all over the world. And I want to highlight the Erasmus University in Rotterdam is one of them. So to promote as many women as possible, not just at MGH, but also within the US and abroad around the world, we wanted to return the favor and now invite women faculty from the host institution that has hosted our Klibanski Scholars to give a talk here together with our scholars in areas that overlap both specialties, so we can foster collaborations across institutions. I think the Erasmus University has been the best. We had several speakers here, and I'm looking forward to visiting you maybe in the summer. And so now it's my great pleasure to introduce our speakers for today. And I will first introduce the speakers, both of them, and then Simone will start, followed by Meenal, and then we will save the questions for the end. So Simone Dalm from the Erasmus University Medical Center in Rotterdam, the Netherlands, is going to be the first speaker. She received her bachelor's and master's degree from the University of Amsterdam and her PhD at the Department of Nuclear Medicine of the Erasmus Medical Center, where she graduated cum laude. She then continued a postdoc in the same department, where she established then her own research line and started her research group, which is called the Radiotracer Interactions Group. In September 2019, she was promoted to assistant professor and received multiple personal industry-sponsored grants, and is also involved in many national and international research projects. And her scientific interests include molecular biology, targeted therapy, and nuclear imaging and therapy. And her talk is on targeted radionuclide imaging and therapy of cancer, novel approaches for fewer side effects and a better quality of life for cancer patients, everything that we wish for. And Meenal Datta is our MGH postdoc, actually. She is a postdoc research fellow in the EDWIN STEELE lab for tumor biology at the MGH and Harvard Medical School. She received her PhD in chemical and biological engineering from Tufts University here in Boston, and her current work lies at the interface of cancer biology, immunology, and mechanobiology in the tumor microenvironment. And her talk is on elucidating and overcoming mechanical forces in the tumor microenvironment. And I just would love to congratulate Meenal, who just received a tenured faculty position at Notre Dame. And according to her, this was because she was part of this Klibanski Scholar and Visiting Scholar program. And the talk she gave and led to her getting a job. So if that is not a success story. So without further ado, I would like to Simone to start our lecture.
Okay, can you see my screen? Great, so thanks for the nice introduction, Miriam, and welcome everyone. So as mentioned, the theme of today's talks is improving cancer imaging and treatment. As you all probably know, cancer is a very complex disease, and there are various hallmarks of the disease, as is also depicted in the figure here, that contribute to the development, the progression, and the invasion of cancer. And in the coming talks, we will discuss with you the research that we do with the knowledge that we have about these hallmarks of cancer and how we use this knowledge to improve cancer imaging and treatment. So how do we use this knowledge? Well, for example, one of the hallmarks of cancer is genome instability and mutation. And this genome instability and mutation causes cancer cells to have a different gene expression compared to healthy cells. This different gene expression causes different expression of proteins or different levels of expression of these proteins. And we can use this protein expression as biomarkers to, for example, identify cancer cells or diagnose cancer. And we can develop targets directed against these biomarkers, for example, for imaging and treatment of cancer, and apply this using novel application strategies for safe and effective treatment. Another strategy is to overcome forces that are present in the tumor microenvironment. The tumor microenvironment is involved in the hallmarks of cancer, for example, avoiding immune destruction and tumor promoting inflammation. And lastly, a strategy is to combine therapies, so a combination therapy strategy to overcome resistance that cancer cells might acquire against monotherapies. So in the coming minutes, I will talk about the strategies that we apply to improve targeted radionuclide imaging and treatment of cancer. And after that, Meenal will talk about elucidating and overcoming mechanical forces in the tumor microenvironment, ultimately also to improve treatment of cancer. So more specifically, as was already mentioned, my talk will be about novel approaches for more cure, fewer side effects, and a better quality of life for cancer patients. So as mentioned, I'm PI of the Radiotracer Interactions Group. Here you see my group members at the moment. And the research line that we study is focused on developing, optimizing, and evaluating novel radiotracers and application strategies by studying radiotracer interactions. And we study these radiotracer interactions on 3 levels. The first one is that we develop and we evaluate novel radiotracers and application strategies. Secondly, we perform studies to unravel the mechanism of action of radiotracers. And with the knowledge that we gain hereof, we try to improve these radiotracers or improve the interaction between radiotracers and their target. And thirdly, we perform studies to identify patient groups that can specifically benefit from the application of radiotracers. So we really look into different subtypes of disease to identify patients that can benefit from the treatments. So what is targeted radionuclide imaging and therapy? For targeted and radionuclide imaging and therapy, we use radiotracers as are depicted here schematically. And these radiotracers are directed against targets or biomarkers that are overexpressed on tumor cells. Roughly, a radiotracer consists of a targeting domain, and via a linker, this is coupled to a chelator. And this chelator is used to attach a radionuclide to the molecule, so to actually make the molecule radioactive. And this is how we deliver radioactivity to the tumor. Depending on the radionuclide that we attach to the molecule, we can either use it for imaging or for therapy. So for imaging, we use radionuclides that have gamma radiation or positron emitters, examples being indium-111 for SPECT imaging or gallium-68 for PET imaging. And for therapy, we use radionuclides that have beta minus or alpha particle radiation, examples being lutetium-177 or bismuth-213, respectively. So what is a very nice thing of these radiotracers is that often we can use the same molecule for both imaging and therapy and just simply switch up the radionuclide that we attach to the molecule. And this is called theranostics. So how did it all start? It started with the first radiotracer that was directed against the somatostatin receptor, which is overexpressed on neuroendocrine tumor cells. So already decades ago, it was reported that the somatostatin receptor is overexpressed on neuroendocrine tumor cells. And back then, there was no cure available for this treatment, and there was also no effective general anti-tumor treatment available. But since the overexpression of this receptor on neuroendocrine tumor cells was reported, ligands were developed that could bind to the somatostatin receptor in an attempt to treat the tumors. However, as mentioned, this treatment was not very effective. Researchers then, actually at a department where I work now, came up with the idea to attach a radionuclide to these ligands, actually to make a radiotracer that could bind to the somatostatin receptor. And the first experiment that they did, this was back in 1980-40, was an autoradiography experiment. What they basically did is they took a fresh frozen tumor tissue from the Erasmus MC biobank from a neuroendocrine tumor. They cut it in thin slices and incubated this with the newly synthesized radiotracer. So what you see is here is a picture of a tumor that was not incubated with a radiotracer and one that was. And everything that you see in black is where the radiotracer bound to the tissue. So what you could see is that they were successful because the radiotracer specifically bound to the neuroendocrine tumor cells. So soon after, many other preclinical studies followed, and ultimately the compound was translated to the clinic. So nowadays, peptide receptor imaging and treatment of neuroendocrine tumors using somatostatin targeting radiotracers is standard of care. And the somatostatin radiotracers were actually also the first that were EMA and FDA approved for imaging and treatment. So following the success of this somatostatin receptor targeting radiotracers, other targeting radiotracers have been developed for targeting other tumor types. And these are directed against towards different biomarkers. And here you see some of them. For example, prostate-specific membrane antigen that is overexpressed on prostate cancer that we target with radio-labeled prostate-specific membrane antigen inhibitors. GRPR targeting radiotracers for targeting of prostate cancer and breast cancer, and radio-labeled fibroblast activated protein inhibitors for targeting of cancer-associated fibroblasts. So here you see a list of some of the ongoing studies in our lab, and I'll give an example of 3 of these studies in the coming minutes. So the first thing that I want to discuss with you is the use of histone deacetylase inhibitors to increase somatostatin receptor expression. As I just mentioned, the somatostatin targeting radiotracers are successfully used in the clinic for imaging and treatment of neuroendocrine tumors. However, when it comes to treatment, although the therapy is effective, complete response in patients is very rare, which indicates that there is room for improvement. So we came up with the idea to use histone deacetylase inhibitors to increase somatostatin receptor expression. So more somatostatin receptor expression on the tumor cells would mean that more radioactivity can bind to the tumor cells. So we would have a higher dose of radioactivity in the tumor, which hopefully would lead to more cell death. So how do these histone deacetylase inhibitors actually work? You can see this in the figure here. These histone deacetylase inhibitors are HDACs, they inhibit deacetylation. And as a consequence thereof, histones are constantly acetylated. When histones are acetylated, the DNA is accessible for transcription factors. So transcription factors can bind, we have transcriptional activation, and this is followed by transcription and translation of proteins, in this case, hopefully the somatostatin receptor. So what we did is we tested a panel of 6 of the HDAC inhibitors to see whether they would indeed increase somatostatin receptor expression. We performed our studies in a neuroendocrine tumor cell line called BON1. And the first experiment that we did is we treated cells with these HDAC inhibitors and then checked whether there was indeed an increase of somatostatin receptor gene expression. And here you see the results. What we saw is that in all cases, for all the HDAC inhibitors that we tested, there was indeed an increase in somatostatin mRNA expression after treatment. Our question then was, does this gene expression actually translate to protein expression, and is the protein actually functional? So do we indeed see that as a consequence of this increased gene expression, there is more protein and there is indeed more radioactivity that is taken up by the cells? So what we did is we pre-treated the cells again with the HDAC inhibitors and then incubated them with the somatostatin receptor radiotracer and determined the amount of radioactivity that was taken up by the cells. Here you see the results. And again, we saw that there was a significant increase of uptake of the radioactivity in the cells after we pre-treated them with the HDAC inhibitors. And for VPA, for example, we saw that this was more than 1,000% higher than when we did not treat the cells. So these results were very promising. And at the moment, we're actually performing cytotoxicity studies to see whether this increase in uptake of radioactivity results in an increased efficacy. So next to the amount of radioactivity that is taken up by the cells, so the dose that is delivered to the tumor cells, an important factor for the success of therapy is also the radiosensitivity of the cells. So how do these cells actually respond to radiation? So what we also investigated is what the effect is of these HDAC inhibitors on the radiosensitivity, if it even has an effect. And to do this, we performed a cytotoxicity assay using external beam radiation after treating the cells with the HDAC inhibitors. And I'll show you the results for one of these inhibitors, VPA. And as you can see, what we saw is that there's actually an increased radiosensitivity after we treat these cells with HDAC inhibitors. So there's possibly a dual function of the HDAC inhibitors. There is an increased uptake of the radioactivity, and there's an increased sensitivity towards radiation. And this altogether would lead, of course, to improve, hopefully, to improved cancer cell death. So as I mentioned, we're currently performing in vitro cytotoxicity studies. And if these are successful, we're moving on to in vivo studies, where we will first perform imaging studies to see whether indeed in an in vivo system, we also see that there is higher uptake of the radiotracer in the tumor after HDAC inhibitor treatment. We then will perform biodistribution studies to see whether this increase in uptake, is it only observed in the tumor and what happens in background organs? And if this is all successful, we will move on to efficacy and safety studies. A next study that I want to show you is a study where we target the GRPR receptor. GRPR stands for gastrin-releasing peptide receptor, which is overexpressed on prostate cancer. Actually, it's overexpressed on almost all prostate cancer. And this, of course, makes it an ideal target for targeting radionuclide imaging and treatment. And in line with this, a lot of peptide-based radiotracers have been developed over the years. And we actually co-developed one of these peptides called Neobomb-1 together with a group in Athens. And we now license the compound to Advanced Accelerator Application, a Novartis company. So here you see the structure of Neobomb-1. It is DOTA coupled, which basically means that we can label it with different radionuclides for both imaging and treatment. And we extensively characterized the molecule in vitro, where it showed that it had very good affinity for its target, the GRPR. It was very specific for the target, and it had an excellent in vivo stability, meaning that it was not broken down by enzymes once we injected it in the bloodstream. So we moved on to in vivo studies, which we actually did in a mouse model of prostate cancer. And we generated this model by subcutaneously injecting animals with the human prostate cancer cell line PC3, which is highly GRPR expressing. And the first studies that we did were biodistribution studies, where we basically injected tumor-bearing animals with the compound, and then at different time points after the injection, we euthanized animals and removed organs of interest and determined how much radioactivity was taken up in these organs in order to follow what happens with the radiotracer after injection. And here you see the results. So we were very happy because we saw a lot of uptake in the tumor. But unfortunately, next to the tumor, we also saw uptake in the pancreas. And this is because the pancreas also has GRPR expression. And we also saw some significant uptake in the kidneys. And this is a consequence of renal excretion of the radiotracer. So although we were very happy, we still have to keep the uptake that we see in the pancreas in our minds, especially if we want to use the GRPR radiotracer for imaging purposes, for example. You can imagine that it would hamper visualization of metastatic lesions surrounding the pancreas. And especially when we use the GRPR radiotracer for therapeutic purposes, the uptake in the pancreas can be somewhat of a problem, because you can imagine that the radiation can perhaps also damage the cells of this organ, which can lead to severe side effects, such as pancreatitis. However, what we did see is that the radiotracer clears relatively fast from this organ. If you, for example, look at 24 hours after injection, which is the orange bar, you can see that almost, I think 60 to 75% of the radiotracer has cleared from this organ. While if you look at the tumor, looking at 24 hours again, you can clearly see that the radiotracer is retained much better in this organ. So after the biodistribution studies, we moved on to imaging studies. So we used the same animal model and injected animals with a radiotracer and performed PET-MR and SPECT-CT scanning, or sorry, PET-CT and SPECT-MR scanning. And here you see the results. We were very successful at visualizing the tumor, indicated by the red arrows in the images. So next we performed efficacy studies, where we treated animals with 3 different doses of the radiotracers and compared this to untreated animals. And here you see the results here of, with on the y-axis, the tumor volume, and on the x-axis, the days after treatment. And if you compare the treated animals with the untreated controls, you can clearly see that treatment was effective. And we actually had a tumor growth delay time that was up to 77 days with the highest dose. And this is also seen if you look at the median survival of the animals, where we saw that control animals had a median survival of 19 days, but this was up to 99 days with animals that received the highest dose of the radiotracer. So at the moment, we're actually performing toxicity studies to determine the safety of the radiotracer. Up to now, we did not see any severe side effects, but the studies are still ongoing. So even though the studies that I showed you were very successful and we're very happy with the results, we went back to the drawing table because of this uptake that we saw in the pancreas. So we're thinking, are there any ways that we can prevent uptake in the pancreas or lower the uptake even, or lower the uptake in some way? Because as it is now, the pancreas is probably the dose limiting organ and would hamper administration of very high doses if you want to prevent, of course, side effects. So we came up with a pre-targeting strategy. And in the pre-targeting strategy, as depicted here, we split the radiotracer in two parts. So what we do is we separate the binding part, the part that binds to the GRPR that is expressed on the tumor cells. We split that part from the radioactive part, and we adapt them both with the click moiety so that inside the body, they can click to each other like two puzzle pieces. And this can be done using click chemistry, namely the inverse electron domain to Diels-Alder reaction is the one that sort of click chemistry that we decided to use. So the idea would be that we first administer the targeting part, the part that binds to the biomarker on the cancer cells. This we injected in the bloodstream. The targeting part would bind to the cells of the pancreas and bind to the cells on the tumor. And then when the binding domain is cleared from the pancreas, but still present in the tumor, approximately around 24 or 48 hours after injection, we then inject the radioactive part that would not bind to the binding domain in the pancreas because simply it would not be there anymore, but it would bind or click to the binding domain that is present in the tumor cells. And in this way, we can deliver cytotoxic radioactivity to the tumor cells, but prevent radiation of the pancreas. So what we did is we made 6 variants based on the mother molecule neobomb-1 that I just showed you. And you don't have to focus on these structures, but I just wanted to show that we adapted the structures in many ways in hopes of finding a very suitable molecule for this strategy. So the first experiment that we did is we checked whether the adaptation that we made to this molecule, whether it influenced the affinity for the target, for the GRPR. So we performed competition binding assays on a human prostate cancer cell line with GRPR expression and compared the affinity of the mother molecule neobomb-1 with the 6 new variants that we developed for this strategy. And as you can see here, there is a shift to the right. So there is a bit loss of affinity, but the affinity of all 6 variants is still pretty good. So the next thing that we did is we tried to evaluate whether this click reaction that we want to happen between the targeting domain and the radioactive effector actually happened. So what we did is we tested them in a tube. So on top you see, on top you see the chromatogram when we only evaluated the binding part. In the second chromatogram, you see the evaluation of only the radioactive molecule. And when we put them together in a tube, you see the result in the third chromatogram. We see that a new product is forming, which actually means that the click reaction was successful. So what we're actually doing now is we're studying other variants as well. So we're synthesizing more variants and testing whether the affinity of these variants for the GRPR is still there and whether the click reaction is happening. We are also performing in vitro binding studies on cell lines and tumor tissues. So we see this click reaction happening in a tube. Will it still happen when we use cell lines that have GRPR expression or use tumor tissue from animals and from humans? And if this is all successful, we'll move on to in vivo studies, where we will first do biodistribution studies to see if there is indeed decreased uptake in the pancreas, and then do efficacy studies to compare our new method with the conventional method to see if it's indeed more effective and safe. So the last study that I want to talk to you about is the combination of radionuclide therapy and immunotherapy. When we administer our therapy, our aim is to, of course, irradiate cancer cells and cause DNA damage. Sometimes this DNA damage can be repaired and the cell survives. But of course, what we aim for is that the DNA damage is such that it cannot be repaired and the cells die. When the cells die, tumor antigens become available, and these tumor antigens can elucidate an immune response. However, cancer cells have a way of evading this immune response by expressing immunosuppressive molecules, which is actually one of the hallmarks of cancer. So what happens is that there is no immune response. But what we want to do is combine immunotherapy with our therapy to block immune suppression so that we will have an immune response and that we will have cancer cells that fire radiation and fire the immune response. So one of these immunosuppressive molecules that is overexpressed on tumor cells is called PD-L1. And PD-L1 can bind to PD-1 and activated T cells, as you can also see in the figure here. And when PD-L1 binds to PD-1, the T cell recognizes the tumor cells as safe and it doesn't attack the cells. So there's inhibition of the cytotoxic T cell activity. And actually, previous studies already demonstrated that there is an increased efficacy when external beam radiotherapy and anti-PD-L1 is combined. And our question is, do we also see this increased efficacy when we combine radionuclide therapy with anti-PD-L1 therapy? So what is actually the difference between external beam radiation therapy and radionuclide therapy? With external beam radiation, an external radiation source is used. It can only be applied for local tumors, and it is a relatively high dose in a short amount of time. However, when it comes to radionuclide therapy, this is a systemic treatment. It's injected intravenously. You can use it to treat localized tumors, but also metastatic lesions. And when it comes to the dose, it's a relatively, compared to external beam radiation, a relatively lower dose for a longer time. So we wanted to know, can we combine this type of treatment successfully with immunosuppression, so with blocking of immunosuppression, sorry, with anti-PD-L1 treatment? So our first question was, do we actually see anti-PD-L1, an effect of anti-PD-L1 or PD-L1 expression after treating cells with our therapy? So we tested this in a prostate cancer cell line, PC3, and we treated the cells with neobomb-1 coupled to a therapeutic radionuclide or with cytokines, and used immunohistochemistry to determine whether, as a consequence of this treatment, there was overexpression of PD-L1. And here you see the results. So we had cells that were untreated, cells that were only treated with the radiotracer, cells that were treated with the cytokines and with the radiotracer, and cells that were treated with the cytokines alone. And what we saw, as you can also see in the figures here, and looking at the pluses, the scoring, you can clearly see that there was upregulation of PD-L1 after our radionuclide treatment. So this is very interesting because this means that if we combine our therapy with anti-PD-L1 treatment, blocking this immune suppression, this could potentially lead to increased cell death. Sorry, so we're actually not only focusing on PD-L1, but we're also checking other immune checkpoint inhibitors such as CTLA-4. We're also checking whether PD-L1 is expressed on other human tumor tissues as well. So we use tissue from our Erasmus biobank to see if there are other tumor types in which we can apply this strategy as well. We're testing different radionuclides and doses to see whether this immunoexpression is, for example, dose dependent. And if this is all successful, of course, we want to translate our in vitro studies to in vivo setting, and ultimately we want to perform combination therapy. So with this, I would like to end my presentation, and thank you for your attention. I'll give the word to Meenal.
Thank you so much, Simone. That was really, wow, eye-opening, fantastic. So much really great stuff. So Meenal, we're going to have you speak, and then please put your questions in the chat, and I will ask them at the end. I already have a couple of questions, some ideas that I got while listening to your talk. So Meenal, please take it away.
Excellent. Can you see my slides here?
Yes.
Okay, wonderful. Well, thank you so much to Miriam and the organizers for inviting me here to speak today. As Miriam explained, I'm kind of now the poster child for this Klibanski Fellowship, so I'm happy to come here to talk about my work today. Thanks so much to Professor Dalm for that very interesting discussion. And as she, suggested, it's really important that we consider these so-called, cancer hallmarks as we think about cancer diagnostics and imaging and treatment. And what I want to speak about today is considering a different facet of these hallmarks, which is what we've started to call the physical hallmarks of cancer. And in particular, I'm going to speak about a mechanical force called solid stress. So as Miriam described in the introduction, I'm an engineer by training, so I tend to think about biology in somewhat simplistic, more systematic terms. And so I'm interested to understand in my research how physical components of tumors can impact disease progression and treatment response, and particularly in the case of brain tumors. And I'll describe a little bit later as to why I'm particularly interested in these very difficult to treat tumors. So I'm a postdoc here at the STEELE labs at MGH. I work under Dr. Rakesh Jain. And in our lab, we study abnormalities in tissue microenvironments that sort of drive disease progression and treatment resistance. And I've been very fortunate to be able to work on a number of different types of solid tumors, including breast cancer brain metastasis, hepatocellular carcinoma, ovarian tumors. And leveraging what we know about cancer, I've even been able to apply these concepts to investigate other diseases, including lung granulomas that result from tuberculosis infection, and benign tumors like vestibular schwannoma that result from genetic conditions. Now, most of my recent work and my postdoctoral work has been focused on glioblastoma, which, as I'm sure many people in the audience know, is the deadliest primary brain tumor in adults. These patients survive less than 2 years typically, and that's because glioblastoma is resistant to current therapies like chemoradiation and surgery. The problem is glioblastoma is also resistant to new therapies like immunotherapy. And part of the reason that treatments fail in cancers like glioblastoma is because of what we call the abnormal tumor microenvironment. And this can really drive disease progression and treatment resistance. So as I'm sure, again, many members of this audience appreciate well, tumors aren't simply a ball of cancer cells. They actually contain all the same biological components as other tissues or organs. But the problem is in the tumor, all of these features are abnormal. So this includes these tightly packed cancer cells or host cells that cluster tightly together and proliferate, and then they tend to overproduce extracellular matrix that sort of holds everything together. Now, blood vessels and lymphatic vessels can be present, but they're often poorly formed and leaky, which really compromises the transport of things like oxygen and nutrients, drugs, and even cancer-fighting immune cells to the tumor site. So eventually, because of all of these abnormalities, hypoxia sets in, and this promotes immunosuppression. So basically, the immune system is unable to fight against the cancer. And in fact, these sort of hostile conditions sort of promote the function of host cells that actually help the cancer cells. So in addition to all of these features, which are sort of chemical and biological, there are also physical facets of this tumor microenvironment that are abnormal. And so this includes mechanical properties like tissue stiffness or tissue microarchitecture, which is basically the maze that cells have to navigate through, as well as mechanical forces like increased fluid pressure, which results from this leaky tumor vasculature, and a force that we call solid stress. Now, we call it solid stress because it's exerted by the solid components of the tumor. So that means the cells and the matrix that they produce. And we call it this to distinguish it from fluid pressure. Now, some of these mechanical features have been very well studied, like stiffness, for example, is already a biomarker of breast cancer, for example. But solid stress is still relatively new. So in terms of our current understanding, our lab has found that solid stress actually promotes cancer cell behavior. And really importantly, it can compress these abnormal and leaky tumor blood vessels. So this further reduces perfusion in the tumor. It can compromise the transport of drugs so that they're not able to be delivered homogeneously. And this, of course, results in reduced treatment efficacy. So you can see that this is a mechanical feature of tumors that actually actively promotes treatment resistance. So up to this point, most of our lab's work in solid stress has been done in tumors outside of the brain, so extracranial tumors, and in the context of standard therapies like chemotherapy. So in my work, I'm particularly interested to understand what's the effect of solid stress in the brain, and can we target it to improve treatment outcomes of new therapies in tumors like glioblastoma, and particularly the outcome of immunotherapies. And in fact, if we think about, what brain tumor clinicians and radiologists look at, I think that they've really kind of long observed and appreciated abnormal mechanics just from standard radiographic imaging. It's just that they gave them different names. So for example, this edema or this excess fluid that sits around a tumor is actually due to this increased fluid pressure. And in the case of the brain, it's due to a breakdown of the blood-brain barrier. And then they'll often observe this mass effect, which is a shifting of major anatomical features of the brain or a compression of those features. And so we propose that that's actually largely due to this forced solid stress. And, unlike in other organs in the body, the brain is housed within this rigid, finite skull. So you can imagine that these mechanical effects are compounded and can be quite debilitating on the neurological health of a patient. So just over the next 10 minutes or so, I'm going to share some of the new approaches that we've recently developed in order to either measure or apply solid stress. I'm going to show you that solid stress can basically wreak havoc on the surrounding brain tissue. And I'll show you just a bit of my ongoing work where I'm finding that reducing solid stress can actually improve the outcome of therapies like immunotherapy for glioblastoma. So of course, the first step is to sort of confirm that the causes of solid stress are there, and particularly in our mouse models of disease. And so when we look at tumor tissues from glioblastoma bearing mice, we find, as expected, a dense collection of cells and matrix, including hyaluronan, which is the most common matrix molecule in the brain, as well as some fibrillar collagens. And all of these can contribute to solid stress. So of course, the next step is to design methods to be able to make high resolution measurements of this mechanical force. So when we think about a tumor that's under solid stress, there'll be areas that are under compression and areas that are under tension. And these areas store the stress in what's known as elastic energy. So you can imagine this as a series of springs that are loaded with weights. And if we were to remove those loads, the springs would relax back to their natural shape. So actually the same thing happens in tumors. So if we release the solid stress by cutting into the tumor, we can relieve that stored energy and the tissue relaxes. And when the tissue relaxes back to its native state, it deforms. So areas that were originally under compression start to bulge, and areas that were originally under tension start to retract. And using optical methods like an ultrasound imaging shown here, we can actually measure the extent to which the tissue deforms relative to the original location that the tissue was prior to cutting into it, shown here by this dashed yellow line. So when we produce sort of maps of this deformation and combine it with the material properties of the tissue, we can actually calculate the solid stress. So this has been a unique application that we've been using in mice, but we've also been able to explore sort of new methods to carry this over to the operating room for patients. So working here with our clinical collaborator at MGH, Dr. Brian Nahead, he uses a system during his brain tumor surgery for patients called BrainLab, which is basically a navigation system. So using this calibrated probe, surgeons can map exactly where they are in the brain back to the preoperative MRI to make sure that they're in the location that they want to be in. So the first step of the surgery is to remove a piece of the skull immediately above the tumor. So, I'm a PhD, so all the MDs know this, but this is what's called a craniotomy. And at this step, surgeons have noted anecdotally that the brain often bulges through that hole in the skull. And so we propose that this is largely due to this mechanical force solid stress. So if we think about our methods of measuring solid stress that I just showed you on the previous slide, when we open the skull above the tumor, we're actually releasing that stored solid stress, and the tissue relaxes, which results in that bulge through the craniotomy. So now in surgery with patients, we can use this navigation probe and this BrainLab software to measure the extent to which the brain bulges through the craniotomy hole. And then from here, just as in our mouse models, we can make estimates of the amount of solid stress on a per patient basis. So this is kind of an interesting avenue that we've been exploring. And what we hope this will allow us to do is establish a patient-specific biomarker of solid stress as sort of a mechanical readout that can be matched in the future to clinical responses. So going back to our mouse models, we know from our previous studies that solid stress is exerted inside the tumor, but now we wanted to know, is it also quantitatively exerted on the surrounding brain? And indeed, using our methods of slicing through the tumor and looking at the deformation, we were able to confirm that indeed there are physical forces that are exerted from the tumor onto the surrounding brain. And since we know from our tumor studies that solid stress can compress blood vessels inside the tumor, we started by looking at the vasculature in the surrounding brain. So I'm showing here a live imaging technique where basically we can map perfuse vessels in the brains of mice through these transparent cranial windows that we install in the skull. So the tumor is outlined here in green, and you can clearly see that this is a very typical, highly chaotic vasculature of a glioblastoma. The blood vessels are highly abnormal. There are areas that are blank, which means that they're not perfused. But we were really surprised to see the extent to which perfusion was affected in the surrounding brain as well. So if we look at, kind of take a closer look, we find that there are many areas with reduced blood flow, areas where blood flow is missing from these compressed vessels. And we even see that this largest blood vessel, the sagittal sinus, is actually shifted over from its original position, as shown here by this white dotted line. So these effects on the surrounding vasculature were pretty profound, and it prompted us to look at neurons, which are in proximity to the tumor as well. And what we found is that these are deformed too. So when you look close to the tumor, they're sort of compressed and squeezed in their morphology. And as you move further away, they tend to look more rounded and normal. I'm not showing the quantification here, but we also found that the actual number of neurons close to the tumor was reduced. So for the purpose of today's talk, I'm not showing the clinical data, but I can just summarize quickly by saying we basically confirmed these vascular and neuronal effects in brain tumor patients. And what this allowed us to do was establish solid stress for the first time as a biomarker, in this case of neurological dysfunction. So the higher the solid stress, the worse neurological performance the patient had. Now, all of these studies were very interesting and encouraging, but they led to a really obvious question, which is how do we know that these effects are actually caused by mechanical forces and not simply from cancer cells, killing off blood vessels or neurons in the surrounding area? So in order to address this, we designed a compression device to mimic the physical features of a tumor. So we took our standard transparent cranial windows that we use for live imaging, and we adapted them and outfitted them with this set screw that we can use to apply the sort of controlled and chronic compression. So by gradually compressing the brain at a rate that's similar to the rate at which a tumor grows, we can actually mimic the way a tumor compresses its surrounding tissue from solid stress. And so this device is really unique because it allows us to evaluate the effects of solid stress in real time via live imaging through this cranial window, and allows us to look at the effects of solid stress without tumor cells being present and exerting other biological effects. So this is a very unique mechanistic tool that we can use just to look at physical forces. So with this device, we again return to looking at the surrounding brain, and using that intravital imaging technique, we basically found that over the time of chronic compression, we see the same effects in the surrounding vasculature close to the screw that we would see when a tumor is present. So vessels become sort of squeezed over time, overall perfusion is reduced. And again, you can see this large blood vessel, the sagittal sinus, becomes displaced and squeezed over time as well. So this confirmed for us that mechanical forces alone can impact the surrounding vasculature and reduce perfusion. So then, of course, we wanted to look at neurons, and we saw the same phenomenon of these compressed neurons close to the screw, and an overall reduction actually in the number of neurons during and after compression, just like when a tumor is present. Now, again, for the sake of time, I'm not going to show all the data, but just to summarize quickly, I can say we were able to use this compression device to confirm that chronic compression reduces basically induces a neurological dysfunction in the mice. And if we decompress the screw, which sort of simulates the surgical removal of a tumor, we can actually abrogate and partially rescue some of these perfusion and neurological function effects. What was fun was we actually used this tool as a drug screening platform, and we found that some agents like lithium can actually reduce this neurological dysfunction during chronic compression. So it opens up some interesting avenues for combinatorial approaches. Now, of course, the question that I'm interested in trying to answer is, can we target solid stress in order to improve therapy outcomes in brain tumors like glioblastoma? and specifically for immunotherapy, because as Dr. Dalm showed in her talk, immune checkpoint blockade, of course, is being tested ubiquitously in cancer, but in glioblastoma patients, it actually benefits about less than 10% of those of who it's tested in. So in order to target solid stress, we basically have two options in this strategy, because there are two components that generate this mechanical force, so either the cells or the matrix. And so in proof of concept studies, we basically found that either depleting cancer cells or depleting matrix could improve perfusion and reduce this physical force solid stress. This is meant to be a combinatorial approach. So by reducing this mechanical force, we can improve the vasculature and perfusion and basically chase it with a drug like an immunotherapy that will be delivered in higher efficacy. Now, unfortunately, these two examples that I'm showing here are just mechanistic. They can't be prescribed to humans. And when it comes to depleting cancer cells, we're not particularly efficient at it. If that were the case, if chemoradiation worked very well, we wouldn't, all be doing the research that we're doing. So instead, I decided to focus on matrix depleting strategies in order to overcome this mechanical force and its downstream effects. And I wanted to focus on an agent that's already approved by the FDA so that it could be sort of rapidly translated to the clinic. And so I was lucky to be able to build on some of the work my lab is already doing. Without sharing today, any of the pharmacological details, I'll just kind of cut right to the chase and say I decided to test a drug called Losartan. This is a drug that's used for hypertension. It's highly safe, it's cheap, it's widely prescribed. And importantly, it can reduce matrix. So our lab has found that this drug Losartan is able to reduce matrix levels like collagen, shown here in a pancreatic cancer model in blue, basically by modulating TGF-beta signaling and other pathways responsible for matrix production downstream. And so you can see here that when we treat this pancreatic cancer model with Losartan, we're able to improve the number of perfused blood vessels shown here in green. And as I mentioned, Losartan on its own isn't going to have any cytotoxic effects. It's meant to be a combination approach. So when you combine this with chemotherapy, you're actually able to reduce these physical forces, improve perfusion, overcome hypoxia, improve the delivery of these drugs, in this case, the chemotherapy, and this ultimately results in better drug efficacy. And so actually our lab has successfully translated this approach into clinical trials for pancreatic cancer patients here at MGH and at a few other sites. So building off of this data outside of the brain, I decided to explore Losartan in the context of brain tumors like glioblastoma and its ability to potentially improve the outcome of immunotherapy. So first, I looked at the effects on matrix and solid stress in these glioblastoma models, and I found that Losartan is able to reduce hyaluronan, again, this very prevalent matrix molecule in the brain, and it reduces the deformation of the tissue after this release of solid stress. So then to test if this reduction of mechanical forces resulted in improved perfusion, which again is the goal of this sort of combinatorial approach, I returned to this intravital imaging technique where again we see the abnormal vasculature of a glioblastoma. We can see the blank areas that are not perfused, and even some areas of the surrounding brain are not highly perfused as well, and this results in hypoxia within the tumor microenvironment. But when we treat with Losartan, we see that perfusion is improved not only inside the tumor, but in the surrounding brain as well. This is confirmed by histology, and we also see a reduction in hypoxia as well. So in my ongoing studies, I've been really excited to learn that this drug, Losartan, has some immune effects as well. So for example, we found that Losartan is able to improve the presence of these cytotoxic T cells relative to regulatory T cells in the glioblastoma microenvironment. And it can also repolarize these tumor-associated macrophages from a pro-tumor phenotype or M2-like to a more anti-tumor phenotype or M1-like. So basically you have more of the good guys when you treat with this drug. And then when we combine with immune checkpoint blockade, so specifically anti-PD-1 antibody, what we find is that Losartan is able to improve the median survival that you would see with the checkpoint blocker alone. And it even induces some of these long-term survivors or mice that have cured tumors. So this is an exciting project. It's ongoing and we hope to publish it soon. of course, there are some questions that sort of remain to be answered that are of particular interest to me, including, sort of what are these factors in the microenvironment that can sort of account for the responses that we see, whether they're biological or physical in nature. So with that, I'll go ahead and wrap up. I'd like to thank my lab, the STEELE labs here at MGH, and my mentor, Dr. Jane, who's been my PhD mentor and my postdoc mentor. We've been very fortunate to collaborate with sort of leading immunologists, single cell sequencing technologists, and brain tumor surgeons here in the Harvard Med network. And we've also been very fortunate to collaborate with mechanical engineers on exploring these physical forces further. So with that, I'll conclude, and thank you.
Wow, that was fantastic, Meenal. Very impressive work. I just put, please put your questions in the chat. I was just typing this. I have not in the chat. Let me, I hope people can hear me too. So I would like a question for both of you. So the pancreatic cancer, terrible disease. There are really no good treatments. Mina, you've shown with the Losartan that there may be an effect, improvement. And Simone, I have a question for you. On your neobone, your marker, you said the pancreatic toxicity was your limiting factor and you're trying to reduce it, but could you use it in your favor and use it to actually treat the pancreas because there was such a high affinity, or image it better? So this was just something that, could you talk more about pancreatic cancer? Okay, well, why don't I open this up to you both? I don't know who wants to start. Simone, why don't you start about this pancreatic activity or affinity? Could you use it for actually to image the pancreas instead of the prostate cancer?
So unfortunately we can't, because what we see is that this target, the GRPR, is also overexpressed in the healthy pancreas, and there is no difference in expression between the healthy cells and the pancreatic cancer cells. However, we are actually performing studies now, and due to the sake of time, I did not show that, where we target the fibroblast activating protein, which is overexpressed on cancer-associated fibroblast. And in the tumor stroma of pancreatic cancer, I think about 90% of the cells are cancer-associated fibroblasts. So what we did is we designed a radiotracer against that could bind to these fibroblast activating protein on the cancer-associated fibroblast. And via an indirect way, we hope to radiate the cancer cells, or at least eliminate these cancer-associated fibroblasts, so we can come in with another treatment that would then reach the pancreas tumor cells and hopefully kill them.
Great, a different approach, but so interesting and important. Well, and Meenal, there is a question for Dr. Datta. What have you seen, what could be the effect of your research in bone cancer? Oh.
That's very interesting.
Thank you.
I can say our lab has not specifically looked at the effects of targeting this mechanical force solid stress in the case of bone tumors. I do think that we have some studies with metastatic breast cancer where we've seen a reduction of metastases to the lung and to other sites like liver and the bone when we combine Losartan with cytotoxic or immunotherapy approaches. I think from a mechanic standpoint, it's a very interesting question, because one of the things that we've learned is that the impact of solid stress within the tumor is obviously affected by the host organ surrounding it. So the brain is very soft. It sort of allows the tumor to do whatever it wants. But in the case of bone, whether it's in the marrow or within the bone itself, the hard, rigid material, I think you could see actually an exacerbation of some of these effects intratumorally, but not necessarily on the extratumor bone. Within the marrow, I think you might see a completely different story. It's an interesting question, one to explore.
What about, this is my area, so we do, I see a lot of sarcomas within bone, but also marrow replacement, like multiple myeloma, for example, which is usually really confined to the marrow space. The bone tumors, they usually go outside. At one point, they break through the cortex. But what about like multiple myeloma or disorders that are really confined to the marrow? Would there be, I don't know, doing some mechanical, could this be a new treatment to do some mechanical, I don't know, burr a hole in the cortex or something like that?
I think it's definitely worth exploring. there is a group, I think, in Seattle who's basically transplanted the bone into the backs of mice and implanted a window above it to do bone marrow imaging. So that could be one application where you could do this sort of live imaging and look at the effects of mechanical forces over time. Marrow is interesting because I think fluid mechanics might play a little bit more of interesting effects there. And there are many groups who are looking at the effects of shear stress and things like that on cancer cell invasion. So many interesting questions.
There is another question here. Do we start the recruitment of pancreatic cancer patients? I assume that was this for the trial?
Yes, so that trial, we completed a non-randomized phase 2 here at MGH. And now we're leading a multi-site randomized phase 2 at MGH and 3 other sites. I believe Dr. Ted Hong is the local physician in charge of the MGH site. Please email me and I'd be happy to provide you with
there are specific questions on the inclusion-exclusion criteria. So why don't would it be okay if I put your email in the chat?
Please do. I'd be happy to send the information that I have, the NCT number and everything.
Let me do this right now, and then we can, okay.
I believe it's for locally advanced pancreatic cancer patients, but do not quote me on that. And they are receiving the MGH standard FOLFIRINOX plus proton plus ICB, immune checkpoint blockade, plus Losartan.
Great, thank you. I have one more question about the mechanics for soft tissue sarcoma. Could there be a role in I saw you're studying schwannomas, which is also really interesting, but I think you talked about vestibular schwannomas, which is again in the brain, but have you looked at peripheral nerve sheath tumors? We're doing a lot of research on those, and it can be actually neurofibromatosis. I just read some whole body MRIs this morning. This can be so debilitating, worse than a malignancy, even if they're so-called benign. They can compress your trachea. You can die from them, from the complications. So
absolutely.
Any words of wisdom from you on benign soft tissue lesions?
Yeah, so we have a professor in our group, Professor Lei Xu, who's actually devoted to schwannoma and schwannomatosis. And she's specifically interested in asking some of these questions about mechanical forces. She's tested Losartan in her models. And one of the interesting things that she has found is that you can actually, one of the mechanisms by which Losartan works in benign tumors like that affect the nerve is it actually basically reduces the pain sensation by reducing fluid and solid mechanical forces on the nerve sheath. So we have a fully dedicated program to that.
Great. Well, and I want to say we should work together, but now you're leaving, but maybe.
Well, Dr. Xu is still here.
Okay. I love this mechanical. Okay, it is exactly 1 o'clock. I would like to thank you so much, Simone, especially coming all the way virtually from the Netherlands.
Thanks a lot for inviting me.
You deserve your dinner now, and it was so nice to hear you, and thank you so much. These were really great and very inspiring and creative talks, so thank you so much, and let's stay in touch. Simone, Meenal, both of you.
Thank you very much for hosting us.
Yeah.
Thank you. Bye.